So while many phages go through a lytic growth cycle, and that is essentially simply how they reproduce themselves, that's not the only type or form of growth cycle that phages can enjoy. And there's a large class, perhaps even a majority of bacteriophages, that enjoy what is referred to as a temperate life state. What that means is that when a temperate phage infects its bacterial host, there are two possible alternative outcomes. One of those outcomes is lytic growth, in which they propagate themselves in exactly the same way as I described to you in the previous slide. And normally that occurs perhaps 80 or 90% of the times that the host cell is infected by a temperate phage. 10 to 20% of the time, there's an alternative outcome. And that alternative outcome is referred to as lysogeny. In lysogeny, the lytic genes, the genes that are required to propagate itself and lyse and kill the cell, they're all switched off, they are repressed, and the phage DNA establishes itself so that it can be propagated in the long term within the subsequent growth cycles of the bacterial host. And that's usually, although not always, accomplished by integration of the phage DNA into the bacterial chromosome. So the phage genome itself becomes incorporated and becomes a part of the bacterial chromosome and gets replicated just like all of the other bacterial genes do. So we can think of lysogeny as a form of parasitism, where the phage is simply going along for the ride in what is otherwise a very healthy cell. It's a free ride through many, many generations of bacterial growth. So lysogens tend to be stable, but they are not they don't have to remain in that state forever. They can go through many, many, many rounds of growth. However, as a process that happens either spontaneously or can be induced by DNA damage, such as UV light, lysogens can leave the comfort of that life cycle, and they can be induced into full lytic growth. There's a relatively easy way to distinguish between phages that are temperate and those that are lytic and can only undergo the lytic growth cycle by the types of plaques that you see on agar plates. Lytic phages form simply just clear plaques, as shown in the bottom right-hand corner here, where all of the cells within that infected area have been killed by phage infection. Over on the left at the bottom here, you can see what temperate phages look like. They form turbid plaques. You see plaques because there's cells that are being killed through lytic growth and propagation of the virus. But there's that important and key subset of cells that in which lysogeny is established, and those lysogens are able to survive and to grow, and they can grow perfectly happy even though they're bathed within this density of bacteriophage particles. So the fact that lysogens can actually grow quite happily, even though there's lots of viruses around to infect them, is referred to as superinfection immunity. Lysogens are immune to superinfection by a phage of the same or a closely related type. This slide just shows an example of how that can be studied and what that looks like in the lab. At the top left here are plaques of a turbid phage, a temperate phage growing on a lawn of bacteria. Using either a toothpick or a wire, you can go and pick cells from the very center of one of those plaques, streak it out on an agar plate, as shown at the bottom left here, in order to generate single colonies, each grown up from a single cell that will have come from the turbid plaque. And then these individual colonies can in turn be tested for their immune phenotypes, as shown in the top right. In this example, we're looking at, we're comparing a non-lysogenic strain with a lysogen. And in the top, we've just spotted on dilutions of a bacteriophage sample. And you can see that these serial dilutions, from the most going down to the least number of particles, gives you the titer, i.e. the number of particles per milliliter of sample on the non-lysogen. But when you compare it with exactly the same dilutions of that particular phage onto a lysogen of itself, so this particular phage is called Giles, on the right is the Giles lysogen, and you can see that there is essentially very little infection, except perhaps a little bit of clearing at the very highest concentration of phage at the top left-hand corner of that panel. And a control phage, in this case below it, showing L5, which is also a temperate phage, but it does not share immunity with Giles, and therefore you see essentially the same number of plaques on the lysogen of Giles and the non-lysogen. And therefore, bacteriophages can be grouped together according to their immune specificities, and it can be done just by comparing the infectivity of a whole set of phages on lysogens and non-lysogens. So this is an important parameter that enables us to group together phages that may be similar by sharing these types of parameters.